super fit. Okay, so what about C then?
Yeah, so if you remember what we talked about with the Human Microbiome Project, the microbial community in the mouse is totally different than the microbial community in the cat. And so
then they interact every time you swallow.
Yes. Yes. Does that make sense?
I should have stopped.
It's okay.
Okay.
Anyway, I appreciate your question though. I'm not trying to trick anybody. So.
Okay.
So that makes more sense. And then probiotics, maybe that's a little bit more obvious example where you're introducing this live microbial community to an existing one, and then the human transplant is similar.
Right. Okay. Should have thought that.
It's okay. Are you have everything set the way you want?
I think so. We'll see. We're recording, so we're on tape.
Excellent. I'm going to look forward to checking out your YouTube channel.
Yeah. It's actually I don't have it on here. I should add it to my list. I keep forgetting. If you just search for Ken Steadman or Kenneth Steadman YouTube, yeah, actually just Kenneth Steadman YouTube I think it comes up. So I can actually check here real quick.
I actually get bored and still listen to it. There's nothing else to do.
You just have it running in the background.
Yeah, usually what
The dulcet tones of my voice. Does it make you really feel mean? Concentrate a lot more. Where are we? So just Ken Steadman YouTube. But I've got a different one. Actually, where's a good place to find that? Let's see. I can email it to you as well.
I'm sure I can find it.
Yeah, I'm pretty easy to find. 1,079 subscribers.
Excellent. How many students have you had?
In those classes since I've done it, it's probably pretty close to that number. But yeah, I probably got a couple hundred from outside. And actually, at one point I got a comment from somebody, I think in Australia, said, "Thank you for posting all your lectures." That's cool.
To look at they probably provide analytics.
Yeah, I'm sure they do. Yeah, let's not bother. We'll just do this.
Wait another minute.
Oh yeah, sure. Let people appear. I'm going to try my super duper pointer thing too.
Oh, that's neat.
Yeah.
So
Well, it's nice because it also records. No, so this is just it knows where I am. So, but it records on here, which is nice. So that's the reason that I use it. Because that's that nice recording. So.
Is your group going to do any field work this summer?
Yeah, we're heading out to Lassen in August, early August. So it'll be fun.
Okay, we'll wait just another minute or so before Professor Steadman gets started. But for anyone who wasn't here on Tuesday, I just want to remind you again to bring 2 copies of your first draft of your article on Tuesday to participate in the peer review process. So there will be points both for the draft and for your participation in the peer review process. And if you don't bring your draft, you can't participate in the peer review process. So I encourage you, remind yourself, print out 2 copies and bring those to class. And if you have any questions at all about how the form that your article should take, just email me or look at the materials I posted on Teach2L. Okay, so I know Professor Steadman has a lot to cover. So this is Professor Ken Steadman. Many of you may have had him for other courses, but I'm really grateful to him for being here today to talk about viruses. So from a true expert. Yes, absolutely.
I just play one on TV.
Yeah.
So.
Without further ado, go ahead. And so Professor Steadman will talk for about an hour, and then we'll do the paper discussion and then the quiz, and we'll try to get them all fit in the class period.
So good morning, everyone. Apologies to those of you who are in virology. You will see some of the same slides again. You may notice my strange hookup here. I will be recording lecture and trying to post it to YouTube. So my standard sort of involvement, but no obnoxious clicker questions and no random roll call questions either. And so of course this is not microbial ecology. It's really all about viral ecology. So, and again, my apologies to those of you who have been in my virology course before, but I don't think you've seen this particular image. 2012, we were warned, climate change, etc. So this brings me to the clicker question that I don't have, but I want you to think about for about the next hour or so. And that is, what do viruses have to do with climate change? A, B, C, or D, and no E because I couldn't come up with anything. Nothing, increased global warming, decreased global warming, B and C. So just as that's a big picture kind of thing to think about for the rest of the class, and then we will come back to this at the end, and then assume we've got any time, you can discuss it with your neighbors and then decide what you come up with. So why do I study viruses? Again, apologies to those of you who are in virology. I don't study viruses for the reason that the vast majority of real virologists study viruses. I study viruses much from an ecological point of view because they're everywhere, they infect everything, they're incredibly diverse, and actually the main reason I got started studying viruses is they're great tools to understand the organisms that they infect. And this is great, it's wonderful, it's an amazing field. There's so much to do, as I will briefly cover in the next hour or so. But unfortunately, what most people get money for is that viruses make people sick, and these are my favorite viral vectors, which I have to show you. Or as one of my students also called them, fomites with feet. And they're appropriately covering their noses at the time. They're considerably older now, but this is a nice, cute picture. The other reason that I'd like to study viruses is this particular one here, which the majority of people in my lab actually work on. This is the SSV virus, and those of you who are in the mutant viruses from hell lab know all about it as well. So ask that person back there a little bit more about the mutant viruses from hell class. But the reasoning here really has to do with the fact that you can understand viruses literally down to their molecular details. And my background is not in virology at all. My undergraduate degree is in chemical engineering, and my graduate degree is in molecular biology. So I'm really interested in how molecules fit together. And so viruses are great because you can actually understand all of the molecules. And one of my colleagues is famous for actually writing the chemical formula for polio, which I won't subject you to here. But at least these extracellular particles have exact chemical formulae, which means that you really can understand them in their molecular detail. So I'll start out talking a little bit about what is a virus, and this is something that us virologists argue about a lot. So this is the standard textbook definition: a very small, infectious, obligate intracellular parasite. Those of you who've had my courses before know that if I put something in red, it means I don't agree with it. And if I put in quotes and in red, it means I really don't agree with it. So we'll talk a little bit about the fact that very small, there are some viruses that have sizes, and I don't know if you've talked about them here at all, but are actually bigger than some bacteria, and some that are almost bigger than some eukaryotes. So the actual virus particle is really quite large. Infectious obligate intracellular, that works. Infectious, goes from cell to cell, only can actually function in those cells. Parasite, again, I'm not so sure about because, as we'll talk about later on today, there are a number of examples that, in fact, virus infection is good for whatever it's infected. And in fact, in some cases, the organism can't even survive if it doesn't have some kind of virus infection associated with it. So parasite, I would say, is not great, symbiont, but symbiont probably going all the way from mutualism to parasitism. What do viruses need? They need precursors, energy, translation, and membranes. And that's pretty much it in terms of viruses. Virus particles are not reused, so if you have a single virus particle, like this guy or this guy, who infects a cell, to over-anthropomorphize here, that's not reused. So if you think about bacteria, you've got binary fission, so half and half. You just keep going that way. In this case, you go from one particle, sometimes more, and then you go to 0 particles, and then a whole bunch of particles. So it's a very different way of replicating, other than what we think about in terms of bacteria. And the vast majority of viruses have a DNA or RNA genome. A couple of them actually have some of both, which is a really interesting prospect as well. So that's the standard, again, textbook definition of viruses with the indications that I'm not too wild about here. This is one of my favorite quotes from one of my colleagues, Patrick Forterre, at the Institut Pasteur in Paris. And ceci n'est pas un virus, which of course is the reference to René Magritte's famous painting, Ceci n'est pas un pipe. So this is not a pipe. Surrealist, but I don't think this is particularly surreal, because what Patrick is really trying to drive at here, almost everybody, when they think about viruses, and this is true for me as well, they talk about virus, they mean one of these extracellular particles. But extracellular particles are not the whole story by any stretch of the imagination, because, I just mentioned, and we'll look at again here, a virus, in order to replicate, has to get into the appropriate environment. What is that environment? It's a different cell. And I just said that this particle is not reused. So what is it when you don't have this particle and you're in the process of making 1,000 more? Is that a virus? Is it not a virus? So that's what Patrick's really driving at here. And so what he would argue, and I try and do this in my virology class, and those people in my virology class know I make mistakes all the time, is when I talk about extracellular particles, talking about them as virions, but the whole replication process as being a virus. So this is much more what I would say that a virus is. You've got this whole cycle, and the cycle here starts where you have one particle that associates with a cell. It puts its genome inside the cell. Then that cell is reprogrammed in order to make more of the virus particles. And then eventually these particles are released from the cell and can go on and do this process again. So I would say this whole process right here, this is a virus, and this is only a virion. And Luria's description of this I really like, from 1978, that they're entities whose genomes are elements of nucleic acid that replicate inside living cells using cellular machinery, specialized elements, these are the virions, that can transfer the viral genome to other cells. The definition of viruses that I use when I'm at OMSI is this one right here. It's a bag of genetic material. And then what Patrick Forterre uses is this one, CEO. So it's not the classic, chief executive officer. This is a capsid encoding organism, because the capsid is what makes the virion. And that is one of the really defining features of viruses. They've got a specialized element that Luria calls it that can survive outside of cells. And so that's that capsid. So that gives you viruses. On the other hand, cells, and I didn't include this image here, are ribosome encoding organisms. So they're REOs. No viruses encode ribosomes. Viruses only encode capsids. No cells encode capsids, but they all encode ribosomes. So way of differentiating between these two. I mentioned this one process, basically what we have here, where we have one particle, releases its genome, makes more particles, etc. This is how people classically think about viruses, but it turns out that probably the vast majority of viruses actually hang out in the environment. It's called this lysogenic cycle, and those of you who had molecular biology know all about lysogeny. Those of you who had virology know even more about lysogeny, but it's the viral genome just hanging out inside the cell, and then when that cell replicates, the viral genome replicates as well. And then you only will have production of these extracellular particles when the going gets bad, or you have to have this kind of production. And this makes a huge amount of sense from the virus's point of view, because if you're killing off a host and you're dependent on a host, that's not a really good way to survive. If you're killing off all your hosts, you're not going to survive either. So this process, the lysogenic process, is probably a much better way to survive in an environment than killing off your host all of the time. So one of the things that I also like to say is viruses have a really bad rap because everybody thinks they do this, whereas most of the time they actually do this. And we'll see some other examples of that a little bit later on. We actually only know relatively recently about viruses. There was just a 100th birthday party for bacteriophage, the discovery of viruses that infect bacteria. And they were originally discovered as disease-causing. Again, a lot like microbes and Koch's postulates. You find something that's really small, causes disease. And in this case, this was after Koch's postulates and after people had shown microbial diseases. And what they'd seen with their microbial disease, and particularly growing microbes, is if you put them through a fine filter, your bacterial culture, you still couldn't see them unless you had Leeuwenhoek's microscopes, you would see no bacteria left that's gone through this filtration. Nothing would grow after it's gone through the filtration, but some of the things in there could cause disease. And so this was really surprising, and people were, well, what the heck is going on here? So this was the very small particles which cause disease, shown pretty independently in the late 1800s by both a Russian group and a excuse me, Dutch group. And originally these were plant viruses. This is the original virus here. Of course, they didn't have electron microscopes in the late 1900s. But this rod-shaped particle, the tobacco mosaic virus, hopefully none of you smoke, but if you know someone who does, and look in a cigarette butt and look at the things you can extract from the tobacco in there, most of the time you'll find these guys. So almost all tobacco is associated with tobacco mosaic virus. Little aside there. Soon thereafter, there were animal diseases which were found, foot and mouth disease, and then these bacteriophage, which were found, again, almost exactly 100 years ago, also independently by two different groups, a British group and a French-Canadian group. And we'll get back to talking about Félix Terrel a little bit later on. So any questions about what viruses are before I move on to the next section? Sure, yeah?
So what in your definition is a capsid even? Because you said no cells can code for capsids. So then is it just something that is like a repeating unit of protein used to make a penogeometric shell or something, or what?
Yeah, so how do you define capsid? And then this gets back to the question that I wasn't going to go into, but I could also talk about for a whole hour, where viruses come from originally. But that the structure, the proteins that make up this kind of capsid or this kind of capsid really don't seem to have any obvious, and again, this is where we could discuss this for a while, homologs in cellular systems. So they really seem to be completely different. Now, that brings up the obvious question about where do they come from? And how do you get a capsid protein? And there are lots of discussion about where that could have come from, but there is nothing inside a cell that even comes close to looking like this. Certainly not at that scale. There's nothing inside a cell that comes close to looking like this. And there are some subcellular compartments, and of course it's biology, there are always exceptions, but for the most part, that works pretty well. The other questions about this? It's when I would normally ask a clicker question, so think yourselves lucky. Okay, so let's talk about the ubiquity of viruses. Viruses infect all known organisms, and hopefully you've seen some variation of this tree multiple times. The big tree, as Norm Pace calls it, the small subunit RNA tree. Yes, you've talked about this probably multiple times. Archaea, bacteria, and eukarya. Of course, we're on this insignificant little twig down here. But every single one of cellular organisms that people have looked at, people have found viruses associated with them. And in the cases where people have looked in more detail, say humans, there are tens to hundreds of different viruses that are associated with them. E. coli is another good example. My favorite organism, Sulfolobus, has multiple different kinds of viruses that are associated. So one species of cellular life, multiple different viruses that are associated with it. those of you who are in my molecular course and maybe in the other molecular course know that 10 to 40% of the human genome is viral in its origin. So not just viruses that infect, but actually have gotten into genomes. This is also true for most bacteria as well. Lots of viral genomes that have been incorporated over evolutionary time into these genomes. Some virus types, and again, a whole other talk by itself, I think a lot like this one down here, which I was very fortunate to be able to be the first person, I think, in the world to ever have seen in the electron microscope. We think there are viruses like this that are probably really old, and viruses are absolutely critical for evolutionary processes. And Patrick Forterre is also one of these people who says that viruses are probably important for the development of eukaryotes in the very first place, and potentially even the invention of DNA. If there was an RNA world before transitioned into a DNA world, quite possibly it was the viruses that came up with DNA in the first place, and the cells said, hey, that's a pretty good idea. I'm going to use that for my genome too. They are ridiculously abundant. If you just take a sample of seawater and stain it with nucleic acid binding stains, you will see lots and lots of dots. Oh, pardon me, went back too quickly here. Lots and lots of dots in it, and those dots, the big ones here, represent the bacteria and archaea. There's one poor eukaryote in here, and everything else, forgot this automatic transition, I should be talking faster, represents all those little spots are viruses, or virions, more to the point. 10 to the 6 to 10 to the 7 per milliliter, which means you have a hell of a lot of them. 10 to the 31, and some people say it's about 10 to the 6 blue whales. If you were to line up all of these particles end to end, they're 10 million light years. And J.B.S. Haldane said that the creator had inordinate fondness of beetles. If there were 10 to the 31 beetles on planet Earth, it would be this big. And there are all these various different viruses lining up. I'm actually out past the Horsehead Nebula. If you just take 10 to the 31 and line them all up next to each other, that's an insanely large number, absolutely ridiculous. So thinking about that is actually very important if you start to think about really rare things that could have happened. So like the development of DNA, or some kind of integration into hosts that change their genomes. Things that happen really, really rarely, if you've got 10 to the 31, even if they happen 1 in 10 to the 20 times, you've got hundreds of billions of these things that are happening. So interesting process to think about. Viruses, and I should say this is not just viruses but also virions, are incredibly diverse. And this gets back to your question a little bit about the capsids. Most virions have very geometric capsids, and those of you in virology know why this is. It's because DNA is incredibly inefficient in terms of coding for protein. DNA is really long, like the DNA in each of our cells is about 2 meters long, so the DNA in any one of my cells is about up to where my hand is. And that needs to be incredibly compacted to get into one of our cells. These virions, for the most part, are extremely small. And packaging their genetic material into one of these small particles is incredibly difficult as well. And so if you think about basic geometry, the largest volume with the smallest surface area is what? A sphere, exactly. And the best way to make a sphere with repeating subunits is something with icosahedral symmetry. So very large numbers of viruses have this kind of icosahedral symmetry just for purely geometric reasons. You also, helical formations, helices are great ways of also packaging nucleic acid. Nucleic acids are helices, but packaging those, so combinations of icosahedral structures and helical structures are what you find very, very often in looking at these virions, and you don't see that in cells. Very, very rarely do you see anything like that. These are your standard viruses. These are some that infect humans, rabbits. This is just an environment. Plant viruses, more human viruses, and that tobacco mosaic virus. One thing that I got really excited about is that the viruses that infect archaea have these really bizarre virion shapes. And we've been working on this one down here, we being the royal we, for actually over 20 years, which is kind of frightening that I've been working on one particular virus for over 20 years. But we've learned a lot in that period of time, and some of the people who are taking the mutant viruses from hell are helping us learn more about these kinds of viruses. But my favorite of these viruses is actually this one here, the Acidionus bottle-shaped virus. This bar, by the way, here is 100 nanometers. So this really does look like a very, very small champagne bottle. You have to drink a heck of a lot of them to get drunk. But this structure is just incredible. How do you get a structure like this at the nanometer scale? And the answer is, we have no clue. So lots of things to work on as far as that's concerned. But it's not just the virions, which are very different from each other. Also, virus genomes are incredibly different from cellular genomes for the most part. Phoenix 174, which is actually the first genome sequence ever to be determined in 1978, 5,386 base pairs. No, I don't think Dr. Thompson is going to ask you that in an exam. But this was so strange at the time. Also had lots of these overlapping open reading frames. So actually one part of the virus genome has 3 different proteins which are encoded by the same nucleic acid sequence. So incredibly compacted. Again, getting back to the idea about nucleic acid is really pretty inefficient. Having all these overlapping open reading frames was, in fact, so bizarre that there was a New York Times article that they thought that these might actually be messages from outer space, which sounds completely crazy. But if you were an alien intelligence and you wanted to send a message to a civilization, or hopefully a civilization at some point on some planet, throwing in a virus which can self-replicate and has these bizarre sequences in it, once they figure out how to sequence nucleic acid, might actually not be a bad way to be sending a message from another planet. So it's not quite as crazy as people originally thought. Part of that, in fact, comes from the fact that these sequences, again, don't resemble anything else that anybody's ever seen before. There are a number of examples here. I'm just throwing up a few of them. Mycobacteriophage. These are viruses which infect mycobacteria. Any of you know about phage hunters or C-phage, been associated with that? It's this absolutely amazing course-based undergraduate research experience. So what students do is they go out and dig up some dirt and then try and find viruses in that dirt in their classes, and then they sequence them, they look at them under the electron microscope, and there are literally thousands of viruses that have been isolated now. High school students, undergraduate students, all studying these different mycobacteriophage. And so literally thousands of them. 50% of the sequences that they find in the dirt samples, wherever you happen to look, turn out to have no similarity whatsoever to any other sequences anybody's ever looked at before. So completely different. As soon as you start to go to a marine environment, it gets even worse or better, depends on your point of view. 91%, and I'll show you the image about this in just a second. I think, did I take out that slide? No, I think I actually took out this slide, sorry. 91% of the sequences you find in marine viruses, kind of like the paper that you'll be talking about later today, don't match any other sequences in sequence databases. So huge amounts of unknown material. And then our favorite extremophile viruses get even more extreme. Up to 95% of the sequences that you find in some of these extremophile viruses are completely unknown. And so people talk about this being, billions of unique genes. And the late Roger Hendricks, who was actually the guy who made all those slides transition really quickly, so it's probably his ghost coming back and making the slides move forward, came up with this nomenclature, the dark matter of sequence space. So as you know, so many of you, dark matter, the matter we can't actually detect is way more than the light matter that we actually can detect. And the same thing seems to be true about viral sequences. Very, very different than any other sequences. And Forrest Rower, another one of my colleagues, at the same time as this paper came out, thought that some ridiculously small percentage of phage genomes have actually been sampled. That was in 2003, and the paper we'll be talking about now in 2016 says, well, actually, maybe not. We're actually, at least for marine viruses, we're getting a lot closer to having saturation of some of these samples. So, what is in these genome sequences? So any viral genome has to have the genes to make more of its genome, has to have the genes that it needs to make a virion, because that's what's defining our viruses. But the rest of them, they actually don't really need. That's about all you need. You need some way of regulating making your genome and some way of making a capsid. And that's the real absolute minimum that you have to have in a virus genome. Turns out that many viruses actually have a lot more genes in them, and those usually have to do with regulating our replication. As I mentioned, we've got lytic viruses and lysogenic viruses. You blast open your host, that's the lytic process. Hang out in the genome. Hang out in the genome is a really good idea. So you want to regulate when you turn on your genes and not turn on your genes. So very often there'll be some of that. Also, how you get from one particular cell to another. And particularly, cells are usually not happy about being blown apart. So often viral genomes will have ways of protecting against the virus infection. And so viruses, not surprisingly, have evolved to have mechanisms for dealing with this. And the classic definition for this of viruses is they don't have genes for protein synthesis, metabolism, or membrane biosynthesis. And of course, quote here, what does that mean? Stedman doesn't like it, because we've now discovered virus genomes that actually do have proteins in them for genes in them for protein synthesis, for metabolism, and for membrane biosynthesis. And I'm not going to talk about all of these. I just wanted to talk about one in particular, and that's this one here, cyanophage. So I mentioned bacteriophages. They're viruses that infect bacteria. What do cyanophage infect? Cyanobacteria, exactly. And these particular ones, I think, are near and dear to Dr. Thompson. Viruses of Prochlorococcus. Really amazing thing was found actually about 15 years ago now, when people were looking in the genomes of some of these viruses that were infecting Prochlorococcus. Many of them had genes that encoded for photosystem components. So what the heck is a virus doing encoding genes for photosystem components? Now that's clearly a cellular gene. Viruses aren't metabolizing by themselves, but they're tweaking host metabolism. And it turns out that there are lots and lots of examples of host metabolism which is tweaked through a virus gene which comes in. Now we're calling these auxiliary metabolic genes. And they're found all throughout different virus sequences, particularly in genome sequences from these marine viruses. And again, those are in your paper as well. Transaldylases, this is a classic metabolic protein. Phosphate starvation and binding proteins also coded for in these virus genomes. So why would a virus genome want to have these? They want to make sure that the host is happy and happy enough to be able to make more virus particles. And it turns out that this infection for the cyanophage allow them to grow in conditions where they otherwise couldn't. And unfortunately, at least for the cyanobacteria, then blow apart the cyanobacteria and make a whole bunch more of these cyanophage. But that tweaking of metabolism we didn't think was something that happened, but again, in the discovery of these viruses of Prochlorococcus, turns out that many of these viruses, actually 88% of the virus genomes actually had some of these photosystem genes in them. Really kind of crazy. So that's, some of the genes that are there, but one of the neat things that you can kind of take this and flip it on its head with, if you now go and look at environmental sequences, again, like the paper that all of you read, I'm sure, for today, we're ready to discuss. You can look at some of the sequences that are found in lots of different places all throughout the world, and in this case, the world's oceans. And what you find is that there are some genes of these viruses that you find all over the place. So particularly DNA polymerase genes. It's a specific viral DNA polymerase gene, actually a viral DNA polymerase gene that's a lot like the viral DNA polymerase gene that you find in this bacteriophage that infects E. coli. You can find in the Southern Ocean, Gulf of Mexico, Arctic meltwater, absolutely identical protein sequences. So you've got crazy amounts of diversity, billions of new genes, but at the same time, you've got some of these which seem to be incredibly successful, and they're spread all throughout the world. So it's an interesting mixture of some incredibly successful viral genotypes, but huge amounts of variability in terms of the much rarer genotypes. And so this is the image that I thought that I'd cut out, but apparently I didn't, of one of those early, this was, 2006, so 13 years ago, studies a lot like the Terra Ocean study that you'll be talking about a little bit later, where they now looked at sequences from 4 different places in the world, the Sargasso Sea, Gulf of Mexico, Bight of British Columbia, and the Arctic Ocean. And the take-home message here is that everything which is in orange, we have no idea what it's infecting, the unknowns here. And then around 10% that are actually known of the sequences when you average all of them together. So mostly unknown sequences. And now we've gotten more and more sequences. And so what turns out is actually most of these orange sequences down here now match to other orange sequences. So we have no idea what they do, but they actually are present in lots of these other viral genomes. And this brings us to our study. And when you do these virus genomics studies, you need lots of material. So standing at the back here is Jeff Deemer, PhD, Portland State University, in my lab. Standing at the front, Eric Iverson, also PhD student in my lab, carrying large amounts of sediment from one of these hot spring environments. So we go and collect our samples. We then extracted the DNA from that. We sequenced it. We compared it to the databases. And what did we find for our sequences? What do you think? Pardon? Lots and lots of unknown sequences, exactly. So 93% of the sequences that we found there were unknown, 7% were known, and again, Jeff really likes this, the whole, dark energy and dark matter end up being 96% of what you find in the universe. So this is, again, really the dark matter of sequence space is what you find in viral genomes. Okay, questions on virus diversity before I move on and talk about ecology? Yeah?
I'm just curious about the environmental compartment.
Mm-hmm.
If you go back to that
Sure.
pie chart.
Oh, which one? Oh, this one here. Yeah.
Yeah.
Hmm?
What's the environmental meaning?
I'm not sure quite follow. The 4 different environments they looked at?
On the legend.
Oh, the environmental. Oh, sorry. Okay, environmental sequences. Sorry, I misunderstood your question. Yeah. So what's the purple, basically? So what's purple is what has already been found in other environments, but wasn't known to have some kind of function. And so,
That environment? Or
so in a metagenomic survey, and I think you've talked probably a little bit about metagenomics here. So people just go out in the environment, pull out DNA, and sequence it. And at this point, not that much of that had been done, at least not for viruses. And so there wasn't very much of the purple. Now, if you were to repeat this experiment, and kind of what the Terra Ocean study did, is probably half of the orange, if not 3/4 of the orange, would now be purple. Because we've done so much more metagenomic sequencing since 2006. Does that make more sense?
Yeah.
Okay. Yeah.
Would we ever be able to determine whether viruses share some common ancestor?
Okay, so question is, do we know if viruses have some kind of common ancestor? Those from virology all remember, what we talked about, common ancestors of viruses? Until we have a time machine, we're not going to know. So that's the easy answer. The other answer is if you start to look at some of the capsid proteins and how those are related to each other, interestingly, you've got some structural conservation of some of these at the molecular level. No sequence similarity, but conservation at the structural level. So maybe, just maybe, that's an indication that you have viruses that are all related to each other. And the best evidence, I think, and I'm kind of biased because I discovered one of the viruses, you have viruses that infect archaea, bacteria, and eukarya that have almost identical capsid structures, just at the protein level, but undetectable sequence similarity.
Could it be convergence?
Again, it could be convergence, but you're getting convergence in bacteria, archaea, and eukarya. Definitely a possibility. Can't rule it out. But another explanation would be some kind of common ancestor. Okay, other questions? Yeah.
These techniques, do they give you lysogenic and lytic virus genomes? And if so, you know, how many viruses, like it's sort of like an estimate for proportion, just never leave the cell?
Yeah, so just to paraphrase your question, and correct me if I'm getting it wrong here, basically these techniques, which I didn't really talk about too much, but these are just virion sequences. What about all of the sequences that are present in cellular genomes? And when you do a metagenomic sequence with cellular sequences, and you find sequences that don't match anything, well, are they viral sequences? How do you know that it was a viral sequence? Or it was a sequence that got integrated into your genome generations and generations ago? So those are a lot harder questions to answer. If it's a recent insertion in the genome, and you can see some sequence similarity, and you've got way more sequence similarity to, again, some of these virome sequences, then it makes sense that that's what it would be. But it's distinctly possible that these are other sequences. So this is all just looking at virions. So if anything, it's an underestimation of the amount of virus sequence diversity which is in our environment. Kind of crazy. But yeah, viruses are crazy, which makes them so much fun. Okay, so wanted to talk about a couple of real virus ecology examples, because after all, this is virus ecology lecture, at least in theory. First one of those is just thinking about marine viruses and what are they doing in the environment. There are ridiculous numbers of them, all those little dots. Those are going to be your virions, presumably, at least in some cases, they're going to be infectious and lytic. What do they do? And depending on how you do calculations, people say about 50% of the death of microbes in oceans is due to some kind of virus infection. That's a really large amount of change. And we'll take a look at this when people do modeling later on. Turns out it's also really useful from our point of view. You probably heard about toxic algal blooms, et cetera. Why do toxic algal blooms end? They die. How do they die?
They make an anoxic.
So that's one reason, but what's another reason? Exactly. The viruses kill them off. So I'm actually just waiting until there's a good algal bloom up in Lake Vancouver, and I'm going to go up and collect some samples and look at them afterwards. But in many cases, these algal blooms are taken out by virus infections. And this is pretty well known for at least marine environments and probably freshwater environments as well. Yeah?
Is it thought that those algal blooms kind of trigger kind of a viral bloom, so to speak, that just subsequently?
Yeah, well, if you think about it, it's a great question. But the idea is that in many cases, and we just talked about right at the beginning, lysis, you kill off your host. It's not such a good thing. But if you've got a ton of hosts around, it's a great idea. You can make way more virus. So it really does seem to be density dependent in many cases. So you got a lot of hosts, you get a lot of viruses. And we'll talk about this a little bit more later on. But this kind of fluctuation in terms of numbers really does seem to correspond pretty well. And so if you think about host availability, and you can just think about like resource availability, all kinds of other ecology systems, exactly the same kind of way. Yeah.
Have there been suggestions or experiments done where people introduce viruses to these toxic blooms to kill it off, and has that gone out of control, or do you think this is a good idea?
So that's an interesting idea. I don't know of anyone who has done something like that, but I know that people have thought about it. And we'll look at an example here a little bit later on, which actually sort of natural experiments that people are doing. And then Dr. Thompson was asking me about phage therapy, which we'll get a chance to talk about a little bit later on as well. And that is definitely a case of where you're trying to introduce viruses to kill off other bacteria. Okay, other questions on this stuff? This is just sort of an overview of thinking about this process, where you have, and you've probably talked about, phytoplankton get eaten by grazers, get eaten by carnivores, and these, of course, are your phototrophs. So this is how you're maintaining a ecosystem, particularly in oceans. But if 50% of these viruses are getting killed, 50% of these bacteria are getting killed by viruses and blasted apart, what this does is it increases the amount of particulate dissolved organic matter in the oceans, which can get picked up by grazers, but also can be used to feed heterotrophic bacteria. And for, I'd say, probably quite a long time, and I think about 15, 20 years ago, you probably know more about this than I do, Anne. People talking about the viral shunt. I think this was probably about 15, 20 years ago that people started talking about this. People didn't know that there were all these viruses in the oceans. And so they were confused because the numbers here didn't add up in terms of your carbon cycling in the oceans. But now if you introduce viruses, it makes a lot more sense in terms of thinking about carbon cycling, or in this case, organic matter. Yeah?
Are there obligate virus predators?
Oh, things that eat viruses, basically? That's an interesting question. I think no one has actually got a really good handle on that. But certainly some organisms can survive on consuming, because they're nucleic acids and proteins, and in some cases lipids as well. So they certainly could serve as a food source. I don't know of anything that actually does just use virions as a food source. But it's an interesting question, and I think people have thought about it before. What I like to think about is sort of in this process, this is a very generalized process, but then starting to think about actual carbon cycling in our atmosphere and in our oceans. And this is from Curtis Suttle, again, about 15 years ago now. If you look at these viruses again, so the viruses, what they're doing is they're contributing to lysis of plankton now. This is both heterotrophs and prototrophs, contributing to dissolved organic carbon in the ocean. So all of this lysis is basically keeping carbon in the upper oceans, and that can then cycle. And that cycling process got lots